Good morning, ladies and gentlemen. It's my big pleasure to be speaker at the US SOT event in Nashville. I'm really sorry that I couldn't join the meeting in person due to several reasons, but I would like to thank organizers for allowing me to present my lecture in this way. So we will be talking about reconstructed human 3D tissue models in toxicology and specifically about their acceptance in the regulations, about how it basically happened that these technologies are nowadays accepted as a full or partial replacement of in vivo testing. Before I will talk about the tissue models, I just would like to mention my expertise that might be of interest in the context of the talk. I have been really actively involved for many years in a number of the validation studies in the area of the topical toxicity testing as junior scientist, as senior scientist, as a manager of the facility that manufactured reconstructed human tissue models, and now also as an academic person working as a director of an institute that deals with the toxicology. So the first studies where I basically came into the contact with the reconstructed human tissue models related to the skin corrosion and skin irritation studies. It was basically part of my PhD work to evaluate different quality of reconstructed human tissue models and to optimize protocols for them, for these tissue models, to allow them to enter validation studies. It was the Skinetic and in some extent also the EST 1000 models that I checked for the possibility to predict skin corrosion. In the same time, I worked as a lab technician and junior scientist in the ECVAM sponsored epiderm skin irritation validation study. This was between 2003 and 2006. And I later on optimized also the Skinetic skin irritation protocol and make it ready for the validation study. I also participated in follow-up validation study for the phototoxicity testing between 2004 and 2006. When I finalized my PhD work at the Federal Institute of Risk Assessment, there was still plenty of work to do with regard to the optimization of the epiderm model for the skin irritation study. So after joining MATEC in 2007, I continued in a work towards the optimization of the protocol and also managed the follow-up validation study with the modified epiderm protocol between 2007 and 2009. Later on, I was also involved in the study managed by my former colleague Julia Kaluzhny that led to the development of the epiocular eye irritation test and its implementation into regulatory guidelines. I also very actively contributed to the development and validation of the epiderm protocol for the medical devices testing that has been implemented into the ISO guidelines. And recently we have conducted together with colleagues from the United States retrospective analysis of the epiderm phototoxicity data sets. That work has been done between 2018 and 2021. So it led also to the implementation of the methodology to the OECD test guidelines. And just before I left MATEC, I was involved in the development and pre-validation of the epiocular time to toxicity test between 2014 and 2018. And as far as I know, the validation is running for the epiocular time to toxicity test. And I hope that also this study will lead to the successful implementation into the new OECD test guideline 492B. So that's basically my expertise and why I am today probably delivering this lecture. Before I will start to talk about the validation studies and how to make the validation successful, it is really necessary to spend time to explain the technology. Because without good technology, without well-developed technology, there would be no successful validation whatsoever. So that's why I have these introductory slides that I would like to share with you to explain you basically that it takes really a lot of efforts and a lot of time to develop technologies that are at the end ready to undergo validation process that itself can take 10 to 20 years. So the 3D tissue models are not really so new technologies. There were reconstructed human tissue models in the market already in early 90s, and they were quite well developed from the very early 3D tissue models. I would like to mention the test skin from Organogenesis or Skin Square from Arotech. And with the Skin Square, actually in Europe, there were the first validation studies conducted. These tissue models, however, became not commercially available around 1995-96, and that was a big disappointment for people that conducted validation studies with these 3D tissue models. Fortunately enough, there were also other technologies, other 3D tissue models available in Europe as well as in the United States, and later on also in Asia, that could basically replace the very early tissue models and be part of the validation projects. So these are the three, I would say, the three key models that went through all the validation studies were the EpiSkin model that was introduced into the market in 1991, the Epiderm model by Matec, available from 1993, and the Skinetic reconstructed epidermis by Skinetic Laboratories in 1994. Later on, around 2000, the commercial producers started also to introduce more sophisticated tissue models on the market. These were the models that contained the epidermis as well as dermis. And of course, other reconstructed human tissue models were commercialized as well. Examples of some of the commercially available skin models you see on this picture. I have only a few of them, and would like to demonstrate to you that all the reconstructed tissue models that are implemented in the OECD test guidelines, which are the epidermis models, they have quite comparable structure, and they also exhibit similar barrier properties. They are not same, but they are similar enough to work more or less under the common protocols that were developed by the laboratories for conductance of the specific tests. As I mentioned, there are also full thickness models available that are usually constructed on layers of either bovine or rat collagen or some alginate based matrices or some proprietary matrices that are covered by the IPR of the companies. More recently, the companies are also exploring a new area, emerging area, that are the disease models. Also here in this area, there is a lot of work that still needs to be done with regard to the creation and development of the models that would be really representing the human skin diseases. As you know, it's not really easy to mimic disease, skin disease, or any disease in the animal models, and this holds also for the skin. That's why so many companies and so many laboratories, also universities or clinics, are trying to come up with a model that would be helpful in researching the skin diseases. Here I would like to mention a new project that has been launched this year. It is a consortium that is organized under the COST initiative. It's called Net Skin Models, and this is a consortium of about 135 experts, mostly from Europe, from European countries, that will try to develop more relevant and more physiologically representative models of the human skin diseases. Let's go to the technology of the tissue reconstruction. So there are basically two ways how to grow reconstructed tissue models, and the one type of the technology is to grow them on the cell culture inserts. The other possibility is to grow on the scaffolds. Both of them are quite well described in the literature, but from my personal experience, I would say that it is a bit more challenging to grow tissue models on scaffolds and make them highly reproducible. So here I would like to show you very briefly how the technology works. Basically, you need to have good cells from the donor. If you are going to reconstruct the human skin, then usually you need for these keratinocytes, if the model should be the full skin, full thickness, then you would also require fibroblasts to have isolated, and maybe melanocytes if you would like to use the skin model for some skin darkening, skin whitening studies. So depending on what you are going to create, let's say we are going to do very simple model based on the keratinocytes. So you take the cells and you seed the cells into the cell culture insert. Cell culture insert is a very simple system, usually constructed from polystyrene, and it has a semi-permeable membrane that can be polycarbonate or Teflon. And through the microporous membrane, there is an inflow of the cell culture media that contains growth factors and some other components that support the adherence and the growth of the cells in the system. So when the cells are feeling well, they are multiplying, and at certain point we decrease the cell culture medium from the system so that the cells are exposed to the air. That type of the cultivation is called culturing at the air-liquid interface, and it helps, this bit stress helps the cells to finalize the differentiation of the layers. They create a stratum corneum, a top, and protect the cell layer underneath. Of course, you need to have also proper culture media and the components in the media that support the differentiation process of the tissue. We have heard a lot in the recent conferences also about the additive manufacturing and 3D printing. This is an amazing technology, and I see a big future in these technologies, especially because these technologies can help us to create scaffolds or supports that have minimal lateral contractions. That is very important for a good reproducibility of more complex tissues. We also hear and see in the literature that the scientific teams are publishing that such 3D printed tissue models may have higher barrier properties, better barrier properties, basically due to the omission of the shrinking of the matrices. And definitely it can accelerate the production process, especially the bioprinting of the scaffold. So what is different in bioprinting compared to the classical technology is that when you isolate the cells are embedded in the bioinks, and the bioinks are inserted in the bioprinter, and you are able to print the layers of the tissues in 1 to 2 steps and create the tissue model in an accelerated manner. Still, for instance, for the skin really needs some time to spend in the incubator to develop into the fully differentiated tissue models. I think it's very exciting technologies, and I hope that we will see in the future much more development in this area. And why we need this variety of the products is quite nicely shown also here. We have some advanced technologies that are available for us scientists these days to advance our research into, I would say, more physiological conditions. Someone said that the models in the incubator in the plastic dishes are bored to death. So we have devices that are stressing them a bit and bringing them into more physiological conditions. On this picture, you have here examples of 4 technologies that I had a chance to see or worked with. There are 2 that are basically relatively simple microfluidic systems. They are enabling the circulation of the media in the system. This is the MIBO device or the Kirchstahl device, and they are basically helping to achieve more physiological conditions in the testing system. Then we have something that is more advanced, and these technologies are called organs on the chips, or even human on the chip. And it is a bit advanced, sophisticated system that allows you to insert the 3D tissue model into a sophisticated chip. You can combine it with several other tissues in one of these platforms. These platforms can be also combined into the larger set, so it's a very interesting technology that maybe in the future will help to address some problems in the area of the systemic testing. These chips or these human on the chip concepts are usually working also with advanced sensors. So, for instance, one of the device that has a nice sensor implemented is the device from Imola that has the tear measurement capacity and enables you to monitor the tissue basically continuously in an automated way. It's not a simple technology, but it's technology that really informs you in a nice way about the microphysiology of the tissues within the time. Let's go back to the reconstructed tissue models itself, because we need to understand very precisely what we are working with, especially in the validation studies or studies that have regulatory applications. This is very important point. So people are asking me frequently what's better to use. Is it the artificial tissue model that is manufactured commercially, or is it better if I do tissues in my own laboratory? Well, it really depends on where we are aiming. If we are developing a new protocol that should aim to the regulatory purposes, I would always go for a standardized model that is coming from a company that has established these tissue models for several years already. They are coming with the certificates of analysis, with the barrier properties checks, with the database of the quality testing, and one can really expect that the models that he or she will be getting for the studies have minimum variability. On the other hand, if we develop a very specific methodology where the commercially available tissue models are not fulfilling our needs, then it is necessary to use homemade tissues. There, however, people really need to be careful what they are creating and whether they are working in a standardized manner, because sometimes they may create big surprises in the laboratory. So the big advantage of the homemade tissues is that you know what's in and you are able to check or add components or materials into your tissues that normally the reconstructed human tissue model doesn't have. You may have a possibility to use multiple donors, especially if you are working closely with some hospitals that have access to the patients. More options and more choices to construct a specific tissue that is available to address specific scientific questions is really of a great advantage. And you, of course, have much more flexibility in planning and conducting studies, because you don't need to wait every Monday for the delivery of your parcel or on Tuesday. So this is, of course, advantage. On the other hand, the creation of the model in your own laboratory may have really some risky parameters. This can be coming from non-donor related variability due to the less stringent requirements for the biological material, less stringent requirements for the cell culture media and the components that are going into the cell culture media, and overall non-GMP adherent production conditions. So that may even lead to some disasters during the pre-validation or validation studies. That's why it is very important to keep good laboratory standards and to follow, for instance, the GMP procedures. So when we are developing protocols where we expect regulatory acceptance, we should really choose materials, tissue models that are highly standardized, and we should really know about the details of the barrier properties, details of the viability, and details on the histology. And how much is still similar and what is different? Well, this is sometimes really hard to say because some of the reconstructed tissue models have some specifics. For instance, more hydrophobic surface or more hydrophilic surface, or a slightly different reaction on some acidic materials. So it's very important to establish good relationships with the tissue model supplier, discuss sometimes the applications that you are trying to develop with him because they know quite a lot about their tissues. And it is important to also collect data on the histology, on the quality testing, and evaluate also your skin model or any other tissue model producers. I do apologize that I always speak about the skin, but I spent most of my time in developing the protocols for the topical toxicity testing. Now let's have a look on the technologies that have been developed for the regulatory toxicology applications with reconstructed human tissue models. So this table very nicely shows some of the tests that are validated and that were implemented into the regulations for the 3D tissue models. One of the first tests were tests for the skin corrosion and later on tests for skin irritation. Also, phototoxicity methods are now in the regulations. They were followed also by eye irritation testing, and in the pipeline we have methodologies that are suitable for the skin sensitization testing, skin penetration, and genotoxicity testing. These methods that are based on the 3D tissue models are not yet implemented, but they are very close to that state. This success was possible because scientists who developed certain procedures were going through very thorough process of the validation. Validation, as most of you know, is a process where we demonstrate reliability and relevance of a particular test or approach or method that are established for a specific purpose. In general, this validation process ensures that alternative methods that were developed by scientists will be accepted by the regulators, regulatory authorities, and the authorities will then be able to use these methodologies for instance, classification and labeling, for approval of the products or for safety testing purposes. Which is what is interesting to know is that most of the successfully validated and regulatory accepted methods were surprisingly developed by industry or as an industrial collaboration with the validation centers. So validation may take a really long time, and in worst case it may take 20 to 25 years until a method makes it from the research and development stage to the final regulatory acceptance. Validation is almost never a straightforward process, and it's going through steps relatively slowly, especially in the phase of the method development and pre-validation. In a pre-validation step, many times it happens that test methods developers have to step back to method development or even research and redesign the whole procedure or the whole model to better mimic the situation that they would like to model in their system. But when the pre-validation is successful, many times people are going relatively quickly through the validation and through the review. Depending on the regulatory agency, the decision whether the method can be regarded as validated and whether it can be implemented into the regulations can be very fast, but it may take again also several years. For instance, at the beginning, it took 4 years until fully validated methods for skin corrosion testing was implemented into the OECD test guidelines. Now this process is much faster, but we have delays in some other regulatory areas, as we know. I would like to speak now a little bit about some specific studies where I was involved and where I learned quite a lot of things that were very useful for other future studies that I was conducting together with my colleagues and friends. My first project in which I got into the contact with the word validation was a project where we validated the epiderm skin model for the skin irritation testing. It was between 2003 and 2006. And I also collaborated with industrial partners in the follow-up validation studies of the modified protocol between 2007 and 2009. So, but let's describe the story. So, the aim of that study was basically to develop a protocol that would be distinguishing between irritating and non-irritating materials. And there was really some kind of specific idea implemented into that validation study, and it was idea that if the reconstructed human tissue models are reconstructed well, if they are similar, they should also be able to use the same protocol or protocol design and the same prediction model. That was a very nice idea, and initially it seemed that it will be working quite well. So in this project, we took a protocol that was developed by a group of Jose Cotovio for episkin model, and we transferred it to the epidermal model without any significant changes that happened in between 2003 and 2004 at BFR. And with a limited number of the materials, we were able really to achieve very comparable results as seen with the episkin model. So encouraged by this result, we felt we are ready to go into the validation study and basically started to test more and more materials in the validation step. Still in the first phase of the validation, it was looking quite okay, but in the second phase where we got to test some mildly irritating materials, suddenly the epiderm model was providing surprisingly high amount of false negative results. So as a result of this validation trial, we learned that the episkin model was considered by the ECVAM as a model that basically fulfilled the validation criteria. It was providing sufficient sensitivity and specificity when compared to the gold standard, which was the rabbit skin irritation test. For the epiderm, there was a really considerable number of false negative predictions. That's why it was recommended to us, to the team who was working on this essay, to redevelop the protocol, the model, or any like a combination of these things in order to better mimic the rabbit data. Of course, we had a look into the causes of this problem because we knew already at that time that the in vivo rabbit data are probably not of the best quality. And indeed, for instance, on this graph that is coming from my PhD thesis, you can see that there is basically no clear border between materials that are classified as irritating and non-irritating. Well, instead of some kind of line that would be discriminating these two groups, you see a kind of gray zone. And in this gray zone, you have number of the materials that are borderline. And if there would be a repeated testing on animals, it is very likely that some of these materials that are, for instance, here classified as irritating would be in the second run non-irritants. So that was one thing that we knew, and where we felt that maybe the epiderm model is performing a bit better than the rabbit with regard to the irritation testing. And we also knew from the published data that there is a large amount of the materials that are irritating on rabbit skin, but that don't irritate human skin. There is a very nice study conducted by David Basketter and his team where they exposed the human volunteers in the 4-hour human patch test. in the same conditions as you would be doing the in vivo rabbit test. And indeed, you can see based on the results and that classification scheme that was implemented around 2006, that 50% of the materials classified as irritating were in fact non-irritating to human patch in human patch testing. So the volunteers didn't react. And when we conducted follow-up study, where we had also again human volunteers against the epiderm model, we have seen the same pattern also in the follow-up study. Materials that were not irritating to epiderm but classified as a rabbit skin irritants were not irritating human skin or very mildly irritating the human skin, which was what is expected basically if you have a well-developed reconstructed human tissue model. So solution for this was not straightforward, because at that time we were really asked to redevelop the protocol in a way to match the rabbit data. So regardless of the knowledge that we had about the causes of this problem, we went the path that was recommended to us and redeveloped the common protocol that it would be more sensitive when used with the epiderm. So we decided to increase the exposure time from 15 minutes to 60 minutes. We also went up with the temperature. We were improving the spreading techniques and washing procedures, and tried to be in fact closer to the rabbits, which was at the end successful process. And we conducted a second validation study with BASF, IIVS, ZBET, and Z Life Science laboratories in between 2007 and 2008. And in this second validation study, we were fulfilling the validation criteria. At the end, both EpiSkin and epiderm models were implemented into the OECD test guideline in 2009 as OECD test guideline 439. So it had a happy end, but we learned quite a lot of interesting information. And the big lessons learned there is that of course, skin models that are similar may utilize a protocol that has similar design, but the protocol really also needs to reflect the barrier properties of a particular tissue model, and we should not really try to fit one protocol for all tissue models, regardless their properties. And the second thing that we learned was that rabbits are really over-predictive models, especially when it comes to the skin irritation and eye irritation testing. And we published on this a very nice paper, which is also quite well cited. And as a consequence, maybe even taking into account these data, the classification system for classifying irritating materials has been changed in the European Union, and the border for the classification was shifted from 2 to 2.3, which was also kind of good achievement. So this was a very nice moment in a Linz Congress in 2007, where we received together with the Czech colleagues and with Dr. Lipsch a very nice award for our work on the human patch testing and comparison of the 3D tissue models to the rabbit trace data. And at that time we were happy that at least scientifically we were recognized as people proving that the skin models can still make a better job than the rabbits. Similar situation that I just described for the skin irritation was also in the area of eye irritation, where we tried to develop a predictive protocol to replace eye irritation testing in rabbits. MATEC had a protocol that was based on the time to toxicity studies, so to say ET50 method, and that protocol was used for many years by the cosmetic companies to predict the eye irritation of the materials. But at that time, the materials that were requested by EQUAM were not looking so well in the ET50 protocol, and it was also seen as a too expensive protocol for testing. So that's why EQUAM was basically approaching MATEC whether we could develop a simplified method with FA-ocular that would address the materials that do not require classification and labeling of eye irritation. It was also because for the severely irritating materials, there were already methods that were validated and regulatory accepted. So there was a need to have another method on the other side of the irritation scale. So it was not an easy exercise, and as you can see, variability of the in vivo data is also great here. This is just a picture from very old study of Whale and Scala showing testing 3 materials in 24 laboratories, and you can see the high variability in the prediction amongst these labs. So for instance, laboratory C is predicting the 3 materials as highly irritating or maybe even corrosive, whereas the laboratory N is predicting these 3 materials as non-irritating. So it's very hard to say which of these prediction is correct, and definitely if we have such a data set, we cannot call this as a gold standard. Nevertheless, this was the situation when we started to develop the protocol. We had to accept the in vivo rabbit data as the gold standard and developed and validate a method against such a bad in vivo data set. So, as I said at the beginning of my lecture, validation is very rarely straightforward exercise, and it was also here in this case, it was not straightforward at all. We learned a lot of lessons in this validation study. We basically started with one protocol that we thought will be applicable for the whole chemical world, but to the end we ended with 2 protocols that are from the test design quite different. The solids protocol compared to the liquids protocol has much longer exposure time, and this is because only with such exposure times we were able to mimic the additional mechanical scratching of the rabbit eye that leads to the enhanced irritation properties of solids. It took us very long to develop such a protocol, and maybe if we would be at the beginning spending a little bit more time in the development phase and in pre-validation phase, it would save a lot of hours to our scientists and also some finances to the validation teams, because we would know that for solids we really need to go to long hours of exposure to get the predictions seen in the rabbit test. Yeah, sometimes it's useful to insist with the upper management that we need to do more testing and training before entering validation. It never hurts to test a few materials more to avoid big surprises later on when the materials after validations are decoded and the data are not looking so well as people would expect. Another problem that we experienced during this validation was a change of the quality of the insert. Basically, it was the membrane that the supplier of these materials changed in the middle of the validation study, and suddenly we have seen inserts that were not resistant to chemicals, and with this testing we suddenly had inserts with the holes in the membrane. The tissues were still intact, they were transparent, they are transparent, so it looks like you would have a hole in the tissue, but not. This is the hole in the membrane that is not transparent. Nevertheless, it really needed to be fixed, so that's why MATEC started to produce their own inserts, and these inserts were even of higher quality and led also to the improvement of the model a bit. And in addition to this technological change during this validation study, we tested also prolonged shipping procedures to the Asian market, to Japan, and also to India, and were able to prove that the lifespan of the tissues on the agar can be increased if the conditions are optimal. So there were a lot of things that we learned here, and we were really happy that at the end we had two protocols that became the OECD test guideline 492. This methodology was later taken on by Skinetic laboratories and also other skin model producers. And we were also happy that our work was awarded at the Linz Congress in 2012 and recognized as a successful project. The last project that I would like to show you, describe you, as a project where we needed to adapt protocol for testing of medical devices. We focus on the intracutaneous irritation testing, and in the area of the medical devices, there were no methods that could address this endpoint at all, despite having some validation studies done for the chemicals. Why that? The word of the medical devices is a slightly different word. Medical devices per se are usually non-irritating materials. They should be non-cytotoxic, non-irritating, no sensitizing. So even if there is something that may have irritation potential, it's usually very low irritation potential. In addition, the board of the medical devices is mostly testing the extracts from the medical devices. So very rarely it is the sample itself that gets into touch with the in vitro system. It's the extract. And this brings additional complexity to such a validation study because the extracts can be prepared by various means. According to ISO 1099312, one can use different conditions for the extractions, and the time of the extraction can take from 24 to 72 hours. So this may bring additional variability, and in addition, this can be also reflected then later on the cytotoxicity. Here we have an example of an extract that is prepared without orbital shaking, and this one is prepared with orbital shaking, and one can see this is a product that is expanding actually with the orbital shaking, but it is also releasing much more of the irritating component into the extract. Going back now to the study, to avoid the problems or variability that this extraction may bring into the validation study, we fixed the extraction conditions at 37 degrees to 72 hours. That was something that was agreed at the beginning of the study. And the second thing in the protocol development was very important consideration of what we have in front of us. So we had materials that have very, very low irritation potential. So if we would use the OECD method that is based on very short exposure time, post-incubation and MTT-assay, that would not really work well. All the samples that we would be testing in such a protocol would be negative. That's why we needed to fine-tune the protocol, and we went towards the protocol that was used, or that is used, for many years in the cosmetic industry, and that works with very long exposure time. In this protocol, 100 microliters of test sample is exposed to the tissues, and the exposure may take from 18 to 24 hours. And in this very long exposure, really, if there is something irritating in the extract, it is usually revealed as a significant drop in the viability. So with this test protocol, we were able to very nicely mimic the intracutaneous irritation testing. You can see how bad it may look in the skin of the animal. There, the barrier properties don't play any more big importance because the material is injected under the epidermis of the rabbit. So if there is something irritating, something that leads to erythema, edema, it is really very obvious in the rabbit skin. With the protocol that utilized the long exposure hours, we were able very nicely to predict this type of very specific irritation with the materials that were kindly prepared for this study by the industry partners. And the whole story from the materials development, from the protocol development, pre-validation, some additional testing that was conducted just before the start of the validation study between MATEC and Riven, and also the story of the kinetic protocol development is described in the toxicology in vitro that was issued in 2018. In 2017 and 19, our work was awarded by the US SOT, and we won an award for the best poster and best paper. And later on, we also received for this work a Lush prize, Lush Award, as a commended project. But the biggest award, at least for myself, was that this method has been implemented into ISO guideline, and in this way, basically, it is a new test method that can be used by the countries that are following the ISO test guidelines, and it can be used to reduce or replace the animal tests that cause a lot of pain and suffering. So my almost last slide is showing you that in addition to skin corrosion, skin irritation, or even photo irritation that we didn't discuss, there are many other applications where the reconstructed human tissue models can be used and will be used. And I think there is a huge opportunity in combination of these 3D tissues with the novel technologies like 3D bioprinting and chip technologies. But it also will require a new generation of scientists that are willing to spend long hours in the laboratories and push for the validation studies and implementation of these new technologies into the regulations. Let me from this place express my big thanks to all the people with whom I had chance to collaborate on these validation studies and validation projects. Without them, we would not have today so many high quality methodologies that can replace and reduce the animal testing. My big thanks are going especially to Dr. Manfred Liebsch and Horst Spielmann, my supervisors from Germany, my former MATEC colleagues, either from Europe or from United States, my supervisor from Germany, Professor Monika Schiffer-Korting, my friends from the Czech Institute of Public Health. Big thanks also to my current colleagues from the Institute of Experimental Pharmacology and Toxicology at the Center of the Experimental Medicine, where I can continue in my work in validating alternative methods. And again, big thanks also to all the industrial partners and associations that were involved in the development and validation of the alternative methods. And with this slide, let me thank you for your attention, and I wish you a wonderful Congress. Thank you. Bye-bye.